This video is your enzyme notes. So enzymes are inside your body, and they are chemicals that are made from your DNA. So the central dogma of biology, dogma means teaching. So the central teaching of biology, the thing that makes biology work, is you take DNA and you code it into this stuff called RNA, and then RNA is made into protein using the ribosomes. And we'll talk a lot more about this process later because it's so important to how life gets made. But the way you do what you do is through what proteins you have. The way you perform actions, the way you have, a certain hair color, the way you have a certain eye color, that has to do with what proteins you have. And all of that can be traced back to what is coded for in your DNA. And these enzymes that we're talking about, these are also in there, in your DNA, because they are proteins as well. And sometimes we don't have the same enzymes. So some of you may be able to drink milk. You go to the kitchen, you get yourself some cow's milk, and you drink it, and nothing happens to you. And some of you may be what is called lactose intolerant. So you drink milk and you get gas and diarrhea, and it's not great for you. And the reason you're that way, if you're lactose intolerant, is because you're missing the protein that lets you to digest that sugar that's in milk. So you're lactose intolerant because you're missing a protein. So we don't always have exactly the same enzymes in our body because we don't all have the exact same DNA. So enzymes are proteins. They are coded for in your DNA. You have a lot of them. You don't need to know the exact number, but you have a ton of different enzymes, and they do so many different things in your body. Enzymes are proteins, but not all proteins are enzymes. So it's not like every protein in your body is an enzyme, but a huge number of them are enzymes. Enzymes are catalysts. So catalysts are chemicals that make a reaction go faster. And so an example of that I often do in my chemistry classes is here. So this is aluminum. The foil is not in there. They're just going to add aluminum foil. And then copper sulfate. And in this reaction, aluminum and copper, they switch places with each other in the chemical reaction. And the aluminum foil turns into copper foil. But if we watch...
Pretty easy to set up, and it happens really...
I'm just going to mute this guy. So he's mixing in his copper sulfate into the water. And then he's adding in aluminum foil here. And then if we watched, nothing's going to happen, okay? It would take a long time, could take up to a day or two to make this foil turn black, to make it turn dark with the aluminum and copper. So these are supposed to be switching places, but you can see that you don't see bubbles, you don't see any hint of a reaction. Nothing's going on here. But we can add something to this reaction here to make it go a lot faster, and that's salt. So here he's just adding regular old table salt, the stuff that we eat, and he's just going to toss it in there. And then the salt, once it starts to dissolve in water, you're going to start to see like little tiny bubbles start to come off of this, and bubbles is often a hint of a chemical reaction, so you can see a couple bubbles there. And the reaction is going faster now, as evidenced by that bubbling there. And so the salt isn't in the reaction. It's aluminum and copper. There's no table salt in this reaction. But what the salt is doing is the salt is just making it go faster. It's making the copper ions switch with the aluminum ions faster and turn into copper foil instead of aluminum foil. And you can see those black dots there. Those are the copper foil. So let's speed ahead about 4 minutes and look at what it looks like after 4 minutes. And you can see that the reaction is nearly done here. The aluminum foil is being replaced by the copper foil. And if left alone, these two would react, but it could take days. But adding the salt just makes the reaction go faster. And that's what a catalyst does, is it makes a reaction that would have gone anyways, it makes it go, but it makes it go a lot faster. Alright, so enzymes are catalysts, like the salt, and catalysts are chemicals that make reactions go faster. Catalysts are conserved, so meaning that if you did a distillation and an evaporation of that reaction that we looked at, you could get all the salt back. You could get all the salt out of that reaction. None of the salt got used up. So in that reaction, the aluminum foil became copper foil, and you can't really get that back, but the salt you could get back. It doesn't actually get used up in the reaction. And the same amount of catalyst that you start with is the same amount that you're going to have at the end. So catalysts don't actually get used in a reaction, they just make reactions go faster. Enzymes are catalysts, but the major difference is that enzymes are found inside living things. So things in your body that make reactions go faster, that are coded for by your DNA, those are enzymes. So enzymes are catalysts, but catalysts are not always enzymes. So your body is full of those enzymes, and most enzymes are proteins. And so there are some examples here. You have lysosomes, which is an organelle in your cell, and they're full of digestive enzymes, and they go around like eating old Golgi apparatus that have died. Lysosomes do that. In your saliva, in your spit, in your mouth, you have amylase, and amylase is something that will turn starches, which are long chains of sugar, into their individual chains of sugar. So if you eat something really starchy, like a cracker, and you chew it in your mouth, you'll notice that over time, as you're chewing it, it gets a little bit sweeter because you're breaking down those starches into sugars. Once your food hits your stomach, it's going to run into lipase, which breaks down fat into smaller fatty acids. And then another one that we're going to talk about later this week too is, as a byproduct of cellular respiration, you make hydrogen peroxide. And hydrogen peroxide in your cells, if allowed to live there in your cells, will kill you. And so you have an enzyme called catalase. Catalase is super important, and catalase breaks down that hydrogen peroxide into water and oxygen. And water and oxygen aren't going to hurt you, but hydrogen peroxide would oxidize your tissues and would kill you. So you break it down really fast, and we'll look at that later this week. Enzymes are very specific for what they catalyze, so just because you have an enzyme, it needs a specific thing that it breaks down. They are always reusable. They never get used up in the reaction. They tend to end in A's, so catalase, amylase, lipase, protease, they usually end in ASE. So sucrase breaks down sucrose, lactase breaks down lactose, maltase breaks down maltose. And what happens is the little substrate fits into a part of the enzyme called the active site, and the enzyme will then break apart that substrate. Enzymes work by weakening bonds, and this lowers the energy that you need for the reaction to happen, and that's called the activation energy. So this is what's called a reaction diagram. You start here with these reactants, and you have to go over the hill, and once you're over the hill, then the reaction can go. But the amount of energy that you put in to get up over that hill, that's called the activation energy. And what enzymes do is they just make it so that the hill is smaller. You have to put less energy in to go down the hill. Okay? So the hill will be shorter if you're using an enzyme. So if a reaction requires the lower activation energy, then it starts quicker, so you have to put in less energy, so they just take less time. And here's a graph of that. So here is a graph without an enzyme. We start here, we got to put in this much energy to get to the top of the hill, and then the reaction can go easily from there. And reaction energy is then released. But the energy that you put in up to the top here, this is the activation energy. This is how much you have to put in to make the reaction go. Then with an enzyme, just the hill is smaller, and so there's less activation energy has to be put in, and so the reaction can get to the end faster because it had less at the start, less uphill time. And notice how you end in the same exact spot. You're never going to get more stuff with an enzyme. It doesn't net you more. It just gets you to the end faster. So enzymes, the enzyme binds to a substance called a substrate. So if this is the enzyme here, this is its substrate, and then they bond together. And so a substrate binds to an active site of an enzyme. So here is the active site. This is where the substrate goes. Usually they're shape dependent as well, so you notice this square fits into that square. So when a substrate binds to an enzyme, that's going to change its shape, and then nothing else can bind while they're bonded together. So let's see if this restarts. No, let me go back. No. So the substrate comes in here, bonds to the enzyme, changes shape, breaks it apart. And that's how enzymes work, is they usually have a shape that fits exactly for their substrate, and while they're doing their job, nothing else can join in that active site. So what are the things that make enzymes go faster or slower? So there's three main things. So environmental conditions are one, and then there's the presence of cofactors or coenzymes, and then there's enzyme inhibitors, things that can turn on and off enzymes. So environmental conditions could be temperature. So being too hot or too cold is the most dangerous to enzymes because that can break them apart. And we'll look at a lab later about that this week. So high temperatures do what's called denaturing, and that unfolds the enzyme. And its folding, its shape is really important to its function. So if it denatures and it gets unfolded, then it doesn't work anymore. pH also affects. pH is like, is it acid or its base? So pH for acid is low pH, pH for base is high pH, and then water's in the middle, it's neutral. So most likely your body is between a 6 and an 8 in your pH. You don't really have a ton of acid or a ton of base in your body. And so that's usually the pH that enzymes work at. And so an example of where that is not true is in your stomach. So if you have enzymes that only work between pH 6 or 8, and you put them in your stomach, they're going to get broken because your stomach has a pH of like 1. So if it has a really low pH, and so you want enzymes that are able to function at really, really low pHs, so that way they can survive in your stomach, and that is the case. So the lipases and the proteases that you have in your stomach right now that break down your food, they work best at really low pHs. So whatever the enzyme, whatever part of the body it works at, it tends to work best at the pH of that particular body part. And then ionic concentrations, so like salt, ions can bind to an enzyme concentration and change its shape. So if you're, usually don't have a very salty part of the body, then the enzyme will work best with low salt conditions. But there are parts of your body that are saltier. So like sweat or tears, the enzymes that are like in your eyes that break down proteins or fight bacteria, they have to learn to work at really salty concentrations. So these are things that can affect the function of your enzymes, but also these are things that could improve the function of your enzymes. If an enzyme is used to working in a salty solution, then it will always work best in a salty solution. All right, cofactors and coenzymes. So sometimes you gotta have your vitamins around to make your enzymes work better. And so those could be inorganic things like zinc and iron, and they can make enzymes work better. So for example, your red blood cells have to carry around oxygen around your body. And so in order to do that, you need iron. And people who have low iron levels, they get what's called anemia, and then you can't carry oxygen around your body as well as you usually do. So iron is, not a living thing, but it's really important for enzyme activity and distributing oxygen around your body. And then you have inhibitors. They can be competitive or non-competitive. So competitive inhibitors are chemicals that look like your usual substrate, but then they try and block the active site of your enzyme. So here's your enzyme. Your usual substrate was like a box, a square, but here's a competitive inhibitor that also has kind of a square in, so it can take that active site and make it so that that enzyme can't bind to anything because it's blocking the active site. And sometimes you want to turn off your enzymes. Sometimes you don't have something to digest, so you want them to chill and stop digesting things. And so you would use competitive inhibitors for that. And now your substrate comes along and it can't bind to this enzyme because the enzyme is blocked. And then there are non-competitive inhibitors, and so they don't go into the active site, they go to another part of the enzyme, but they cause the enzyme to change shape, and now the active site doesn't look like it used to. So here's your enzyme. A non-competitive inhibitor comes back here, and now instead of having that usual square shape, it's kind of pointy. So when the substrate comes around, it can't bind there anymore because it used to be a square, and now it's not, and so the substrate doesn't fit there anymore. So that's non-competitive. They go to a different part of the enzyme, not the active site. All right, and that's it for your enzyme notes.